Bismillahirrahmanirrahim. Assalamu alaikum dear all students. Welcome to the lecture 10 of histotechnics. This lecture is about the next step after the tissue processing, that is the embedding of the tissues in the molten paraffin wax. The outlines of this lecture is first the aims of embedding, what is the purpose of embedding, and then the different types of embedding media and the different molds that is used for the embedding of the tissues. What is the embedding process, how a tissue is embedded in that embedding medium, and in the last we will discuss the machine which is used for the embedding of the tissues with the paraffin wax. Embedding is a process in which a tissue specimen is surrounded with a molten medium by using a specific mold, a specific designs, and after that the medium is solidified so that a block is prepared, and this block helps in cutting of the thin sections to prepare the smaller section of the tissues to examine and study under the microscope. The aims of embedding medium is to give support to the tissue, and the second is to prevent distortion of the tissue during cutting. So, as the tissue contain different empty spaces which contain fluid or water, so that empty spaces is filled with this embedding medium, and this helps in the maintenance of the structure of the tissue and supports the tissue, and also this prevents the distortion of the tissue during cutting, that if we apply blades and it is a pressure is applied on the tissue for cutting purpose, then that tissue do not disturbs and their structures are maintained. The third aim of the embedding is to preserve the tissue for archival use. So, if any tissue is embedded in a medium, then we can put this tissue, we can place this tissue for longer time and for study purpose or for the reposition in the library for the future use or the future research. Now, how we can select the embedding medium? The different types of embedding medium are available for this purpose, and the one of them is the paraffin wax, the epoxy resin, the methacrylate, carbowax. So, these are the reported different types of embedding medium, but the paraffin wax is the most commonly used embedding medium throughout the world, and it is the non-toxic, cheap and easily available from the crude petroleum. It is a byproduct of the crude petroleum. The choice of embedding medium, it depends upon the type of tissue, that is the density of the tissue, and the embedding medium, it should be close, otherwise tissue may not be sectioned properly and the tissue will be deformed. So, the type of the tissue is important during the selection of the embedding medium. And then the type of microtome for sectioning of the tissue, it also matters for the selection of the embedding medium. and the type of microscope, normal routine microscope or the electronic microscope, or the electron microscope. So these are the factors which affects the embedding medium. So embedding medium must be selected after looking for the type of tissue, the microscope type and the microtome type. Paraffin wax is the most commonly used, and it is a solid polycrystalline hydrocarbon, and this paraffin wax is sold in the market with different melting point. Their melting point varies from 39 degrees centigrade up to 70 degrees centigrade. So this is the variety within a simple paraffin wax. These paraffin wax are cheaper, they are cost efficient, and they are very easy to use. And for working with the paraffin wax, a little supervision is needed during the blocking and the block preparation of the tissues. The next embedding medium is the epoxy resin. It is mainly used for the electron microscopy, and as we have already discussed that the three factors is important for using the embedding medium. Epoxy resin is used for the electron microscopy because it provides better resolution and greater details of the tissue. But the issue with the epoxy resin that it is costly than the paraffin wax, and paraffin wax do not give the details that is given by the epoxy resin. That is why for electron microscopy epoxy resins they are used. Acrylic medium is another embedding medium, and the methacrylate monomer, that is the main part of the acrylic medium, and this is soluble with the ethanol. So in the presence of benzoyl peroxidase, which is 2% in the concentration, the methacrylate monomer is polymerized, so they provides a hard and the clear block. The agar gel, which helps in the cohesion of breakable, friable and fragmented tissue. These particularly used in cytology sample, endometrial keratin and small endoscopic biopsies. This gel do not provide support the tissue for sectioning. So agar paraffin wax double embedding medium is used, and this complex is more suitable for the agar alone in the embedding of the tissue. Gelatin is the next type of embedding medium. This is used in small friable tissues and also for the frozen section, which may contain a friable or necrotic tissue. So for that purpose gelatin is used. The melting point of gelatin is 35 to 40 degrees centigrade. So due to this melting point, the gelatin is not used routinely for the embedding, because in certain areas the room temperature is around about 35 to 40 degrees centigrade. So this is difficult to maintain the tissue in the solidified form. So that is why this is the major disadvantage of gelatin. The celloidine is another type of embedding medium, and this celloidine is basically a nitrocellulose, So, this celluloid medium is used for embedding of the hard bony tissue, for example, the bones, the cartilage, the eye specimens. But due to its several disadvantages, this celluloid medium is not used in the labs nowadays, and paraffin wax is preferred over this celluloid medium. The different type of molds, the different type of structures that is used for blocking is, the first one is a stainless steel mold. This is made of stainless steel and the base of the mold is flat and well polished, which helps to remove the liquid paraffin. Here is the structure of the molds. These molds are then covered by the plastic ring. So, these are of different sizes, and this is a medium type of mold. They can be smaller than this one for smaller tissue, and for larger tissue the mold use is slightly the base is slightly larger than this area. And the plastic mold, here is the example of plastic mold. So, the mold is made from a plastic which is chemically resistant, so that it may not react with the chemical substance that is used during the tissue processing, or it may not react with the medium which is used for the embedding process. Another type of mold, that is leuchard embedding molds. This is a type of embedding molds which have two arms. The one arm of the L is longer and the one arm of the shorter. The two L-shaped arms are adjusted to make a convenient size of the block, just as we can see that this is the two L-shaped arms, and they are adjusted according to the convenient of the tissue. Before embedding the tissue in this leuchard embedding molds, first lubricant is applied, for example glycerin, to the arms of the molds and metal plate, so that they should be easily removed and the tissue may be easily separated from these molds. The molten wax is poured in the space between two arms, and then the tissue is placed in it. After pouring and after putting the wax in that, they are cooled and the block with the tissue in one surface is removed with the help of microtomy. A system which is used for the embedding is the tissue tag system. This system is available and it can be used for all types of embedding medium. The things which are required for the tissue embedding is the molten paraffin wax, which is the embedding medium, and the mold, either it may be plastic or it may be of metal. The metal plate, cold plate, so that it is used for the solidification of the liquid. And this is the picture of a tissue embedding tag system. This is the display which shows the temperature. Here a wax dispenser in which the wax tank is present. So, it dispenses the wax within the molds. The forceps warmer, which is used during the dispensing of the wax. The hot plate, which is hot from the lower side, so it makes the wax hotter. Then we can adjust the tissue as according to the dimensions that is required for the microscopy. This is the tissue warmer area, so tissue is placed in this, and after putting the wax in the molds, then they are placed on the cold plate so that the wax solidifies and we can easily separate the molds from the tissue. The tissue tech system, multiple steps are performed by this tissue tech system for embedding of the tissue. First, the liquid paraffin, it is kept at constant temperature in the dispenser in which the liquid paraffin is present. Then the tissue is put on the lower surface of the mold with the help of forceps, and the tissue is placed correctly so that all the surface of the tissue they are appear on the smear. The cutting surface of the tissue is directed downward, and the molten paraffin wax is poured on the metallic mold. So, after pouring the molten paraffin wax in that mold, then it is covered with a peripheral plastic ring, and after covering it is labeled. Then the mold is put directly on the cold plate so that it gets solidified and the tissue is fixed and it is enclosed in that molten paraffin and it is embedded in that. When the tissue gets solidified, the metallic mold is detached from the plastic ring and the block is used for the cutting of the tissue. This is the image of the tissue tech system. This is the whole system of the tissue track. First, after fixation of the tissue, it is taken out from the wrapper or from the container or from anything in which the tissue is present. After that, the molten paraffin wax is poured on the mold. Here we can see the largest size mold in which the tissue is present. So, then the tissue is embedded in paraffin and the tissue is set with the help of warm scalpel or forceps. Just we can see here that the tissue is forced downward and the cutting surface is on the lower side of the mold. Then the tissue is pressed on mold so that the cutting surface moves to the down surface and it faces the blade of the microtome. After that, the mold is covered with a plastic ring or a plastic cassette. This plastic cassette is made from a high quality plastic so that it do not react with the chemical substance or with the molten paraffin wax, so that it changes the chemistry, the chemical properties of the paraffin wax. Therefore, it is made from such a plastic that this cassette do not react with the paraffin wax. Then unique number is put in the plastic ring and then the molds are put on the cold plate so that it gets solidified, and after when the paraffin wax solidifies, then the molds is removed and the extra wax that is also trimmed with the help of a blade of the microtome. So this is the whole system, the whole process of the tissue tech system. That was all for the embedding of tissue. Hope you get enough knowledge about the embedding of tissue. If you have any question, you can ask through the question box. Thank you. Assalamu alaikum and Allah Hafiz.